Today, we're diving into the fascinating world of genetics, exploring how offspring inherit traits from their parents. We'll predict offspring ratios by examining various inheritance patterns, including dominant and recessive alleles, and how these contribute to genetic diversity. A Punnett Square is a graphical representation of the possible genotypes of offspring arising from a particular cross or breeding event. Creating a panicked square requires knowledge of the genetic composition of the parents. The various of possible combinations of their gametes are encapsulated in tabular format. Therefore, each box is a table represents one fertilization event. The inherent assumption is that each trait is determined by a single gene locus and that various traits assort independently from one another. Though this is true for many useful traits, especially when choosing characters for plant or animal breeding, there are many exceptions. This tool was created in the 20th century, much after Mendel's seminal experiments on genetics. However, they are now commonly used to explain the results that Mendel obtained, especially when combined with our current knowledge of DNA, genes, and chromosomes. A Punnett square is our geneticist toolkit for predicting the possible genotypes of offspring. Like a chessboard, each square is a potential move for our genetic game, combining one maternal and one paternal allele. We'll begin by identifying the parents' genotypes and align them on the top and side of our square. As we fill it in, we visualize the dance of inheritance, the meeting of each egg and sperm, and capturing the essence of genetic shuffling. The genotype of an organism is the chemical composition of its DNA, which gives rise to the phenotype or observable traits of an organism. A genotype consists of all the nucleic acids present in a DNA molecule that code for a particular trait. The outward appearance, or phenotype, is the result of interactions of proteins being created by the DNA. Modern DNA analyzing techniques have made it easier to identify each segment of DNA which are responsible for various phenotypes. A genotype has different alleles or forms. The different alleles are produced by mutations to the DNA and may give rise to beneficial or detrimental changes. In bacteria, the DNA exists in a ring and only one allele for each genotype is present. Sometimes an allele will mutate in a beneficial way, the organism will reproduce more, and the genotype will increase in the population. In sexually reproducing organisms, there are two alleles present in each organism, which can have complex interactions with each other and other genes. Mutations can occur in these alleles, new combinations can arise during meiosis, and infinite amount of variety can be created. These combinations of genotype give rise to the enormous variety of life on Earth. Example, eye color. Although a particular genotype consists of many nucleic acids, scientists typically represent genotypes with single letters, or two letters in the case of sexually reproducing organisms that receive one allele from each parent. For example, the trait for eye color could be represented with the letter E. Varieties or alleles of that trait that are dominant will be designated by capital letters. Therefore, E will represent brown eyes. Traits that are recessive are written in lowercase. The allele for blue eyes is recessive to the allele for brown eyes, so we call it lowercase e. A set of parents has brown eyes. Having brown eyes only tells us that phenotype, not their genotype. The parents could be capital letter E for both, or capital letter E and lowercase e. Or there could be one of both. A single E allele in the genotype will result in the brown-eyed phenotype, even if the parent harbors a recessive E allele as well. The parent conceives a child. The child has blue eyes. This tells us that the child is homozygous recessive, or lowercase e for both cases, because only two recessive alleles can produce blue eyes. This also tells us a lot about the parents. The baby having two lowercase e got one from each parent. Therefore, each parent has one lowercase e allele to give, while having the brown-eyed phenotype. This shows us that the parents have the heterozygous E genotype. If either parent were homozygous dominant, the baby would have received at least one dominant E allele, giving it brown eyes. In the case of cystic fibrosis, for a long time it was not known why some children would develop a thick mucus in their airways, causing them to be short of breath and wheezy. The children have a variety of other symptoms, each as the inability to process food efficiently, gas, and weight loss. Until recent advances in medical and genetic sciences, many children died at a very early age. After years of research, cystic fibrosis was found to be caused by a defect in a gene that produces salt channels across cell membranes. These salt or ion channels are used to maintain pH levels in various cells, remove wastes, and remove nutrients from their intestines. The genotype of people with cystic fibrosis is homozygous recessive. In other words, they carry two copies of the non-functioning allele for the gene that creates specific ion channels. For some people, known as carriers, can have a functioning normal phenotype while having a heterozygous genotype. This means that a carrier can pass a non-functioning allele to their child. Unknowingly, two carriers can both pass on the non-functioning allele, and the child will receive a non-functioning homozygous recessive genotype. However, if one or both carrier parents passes on their good allele, the child will not have the symptoms of cystic fibrosis. If the child receives one functioning and one non-functioning allele, they will be a carrier as well. The child will not be carrier if they receive two functional alleles. However, in parents who are both carriers, the genotypic ratio of offspring will be set at a 1 normal and 2 carrier and 1 cystic fibrosis genotype. This genotypic ratio can be counted in a Punnett square. Place the heterozygous parents on the sides of the square, separating their individual allele. A and lowercase a, uppercase A, lowercase a. So you will have there uppercase, uppercase. So this will be a normal person without cystic fibrosis. Uppercase A, lowercase a. So this is a person which is a carrier. This is a person which is also a carrier. And this is a person with a functional allele which is cystic fibrosis. It can quickly be seen by counting that the genotypic ratio is 1 uppercase A, both uppercase, 2 uppercase and lowercase a, and 1 lowercase and lowercase a. And in this case, the phenotypic ratio would be 3 normal. Phenotypic ratio, phenotypic ratio is 3 is to 1. The genotype is the underlying genetic script of life. This script, when written in the language of DNA, directs the creation of proteins, which are the actors on the stage of life, contributing to the phenotype, the traits we can see and measure. With advancement in DNA analysis, we can now trace back the scripts responsible for everything from eye color to enzyme function. To unravel the narrative of genetic inheritance, we turn to genotypic ratios. Using a monohybrid cross in our Punnett Square, we will see how often a characteristic appears in the next generation. It's like a genetic forecast telling us the likelihood of each genotype arising from a pair of alleles. Let us make a Punnett Square. We are going to use a heterozygous parents for the color of the flower, as the one that you see here in the picture. So here are the alleles for the parents, capital P, lowercase p. So let us make the cross. So we will have one homozygous purple, and then we are going to have heterozygous and a homozygous, which will produce white. The genotypic ratio shows the number of times a characteristic of an organism will be seen in the offspring when genes for certain traits are crossed. This more easily understood by using the Punnett Square method and a basic monohybrid cross. The image shows a Punnett Square monohybrid cross of a male and a female pea plants that are both heterozygous dominant for purple. The genotypic ratio for this cross is written as 1 is to 2 is to 1. In animals and plants, each gene has 2 alleles or variations, one from each parent. When male and female gametes come together, all the phenotype variations for the offspring are predicted using the Punnett Square. The pairs of alleles for male and female are plotted individually on a grid, and then each square in the grid is filled with the corresponding combinations of alleles combining from the parents. To find the genotypic ratio, count the number of times each combination appears in the grid, starting in the upper left square. The example below is crossing alleles for just one trait, flower color. Larger Punnett squares are used to calculate genotypic ratios for more than one trait. This is, in our case, this is the one that we will be focusing on, a monohybrid cross. A phenotype is the physical expression of DNA. In contrast, the genotype is the chemical makeup of DNA that causes a particular phenotype. DNA is first transposed into RNA, a slightly different information molecule, which can then be translated into a protein. Phenotypes are caused by the interactions of many different proteins created by DNA. This process can get incredibly complicated. Even in the lowest life forms, there are thousands of genes creating thousands of different proteins. These proteins interact in complex ways with each other and the environment, allowing the organism to obtain energy from nutrients. In simple sense, each gene can produce a single protein. This protein can make several different forms based on the different alleles in the population. Some of these forms will be functional, while other forms will be less functional or not work at all. If a trait is controlled by a single protein, the genotype that the animal receives for that protein will determine if that trait is adaptive or detrimental in the pursuit of reproduction. For instance, people with cystic fibrosis have a mutation in the gene that produces a protein specific to transporting chlorine molecules across membranes. Because their genotype contains a non-functional mutation, the protein created does not function properly. Thus, they have a non-functional phenotype causing many health problems. Example of phenotype: melanin. Melanin is a molecule produced by many animals. It is known as a pigment or a molecule that gives tissue a dark color. In humans, varying levels of melanin in hair, eyes, and skin are what cause such a wide variety of appearances in the world. While there are many genes that could control the distribution of melanin, there are only a few genes responsible for its production. That is why in many populations around the globe, individuals can be found that produce no melanin at all, a condition known as albinism. Those with albinism, regardless of their race or parents' phenotypes, lack the ability to produce melanin. Without the pigment in their system, their hair and skin appears white and eyes usually a shade of pink. All populations contain albinism because the pool of genes responsible for producing melanin is large, and a mutation in any of them can disrupt the entire process. Although mutations are rare, when enough people are reproducing, the trait will appear. However, because of the decreased protection from the sun's rays, many with albinism are subjected to a variety of skin and eye cancers. This may explain why albinism is usually seen in only a small portion of the population. Animals also experience albinism, as the pathways that produce melanin in them are much the same as in humans. In fact, albinism has been documented in most mammals. All mammals use melanin as a pigment. Other groups of animals use different pigments with different mechanisms for making those pigments. Albinism is also possible in those animals if the genes that produce the pigmented phenotype get disrupted. Sometimes these mutations are selected for, as in the case of winter animals, which often show partial albinism in their coats, allowing them to blend in and absorb more solar energy. Let's go back to Mendel's piece. Gregor Mendel is most known for his contributions to genetics. As a friar at Germany Abbey, Mendel studied the phenotypes of peas. Mendel was particularly interested in the phenotype ratio between the offspring. Mendel noticed that when he bred yellow peas with green peas, sometimes the offspring would be half yellow and half green. and sometimes the offspring would be all yellow. Mendel concluded that each pea carries two forms of the gene for color. We now call this alleles, and we know why Mendel's peas would produce offspring in different ratios of colors in the Punnett squares. Each square represents a potential offspring between the parents listed on the outside of the larger square. Remember, each parent gives one allele to each offspring. The Punnett square is easy way to visualize this. The phenotype of color in peas is caused by a gene that produces the yellow pigment. Without this pigment, the chloroplast in the pea pod give it a green color. Each organism contains two alleles for the gene, one recessive or one received from each parent. If an organism receives even one allele that can produce the yellow pigment, the pea pod will be yellow. If, however, neither of the alleles can produce the yellow pigment, the peas will appear green. The yellow allele is known as the dominant allele because only one will cause the yellow phenotype or outward appearance. The gene allele is recessive allele because you need two green alleles in an individual to produce. Phenotype is the physical expression of the genotype's instructions. By looking at our guinea pig examples, we'll predict the blend of traits in their offspring. The phenotypic ratio is our calculator, giving us the expected frequency of each trait based on strict genetic accounting. The phenotypic ratio helps us to predict gene expression in the future generations of organisms. In phenotypic ratio calculations, we map out specific parental alleles and predict the probability of how they will be expressed in their offsprings. Knowledge of allele dominance is required, although it is possible to figure out very simple parental genetic makeup by looking at observable traits or phenotypes in their young. To conclude, predicting offspring ratios through various inheritance patterns is a journey through genetic possibilities. From dominant and recessive interactions to the understanding the nuances of codominance and incomplete dominances. We have the tools to foresee the rich tapestry of life's potential traits. Now, let's put this knowledge into practice and explore some genetic scenarios together.